Good morning, class. So today our topic, of course, you have PCR or polymerase chain reaction. And so usually yung PCR, we have three important steps. We have denaturation, annealing, and of course, extension. So important, of course, mag-start tayo with template DNA. And of course, our template DNA, double-stranded siya. And along here, along the nitrogenous bases, we have, of course, hydrogen bonds. So in the process of denaturation, we try to increase the temperature until until such time na mag-absorb ng energy yung hydrogen bonds at ma-break yung hydrogen bonds para ma-separate yung DNA strand. Okay. So, pag na-separate yung DNA strand, we try to lower down the temperature until such time that our primer, you remember in microbial genetics, we have RNA primer, which is very important to initiate yung action ng DNA polymerase para maglagay ng newly strand DNA. Okay? So, annealing is important part because ito yung time kung saan yung primer is mag-a-attach dun sa target gene natin. Okay? Target strand. Okay. And then after that, we change again the temperature. Now we have extension process. So in extension process, we allow our um, itong ating DNA polymerase to make yung complementary base pairing para makabuo ng bagong strand. Okay. So mag-repeat lang lagi yung cycle. So this is the first cycle. So if you have one copy of DNA and then after first cycle, meron na kayong 2 copies of your DNA. And then second cycle, you have 4. So ito ay exponential. So kung maraming cycle, 30 yet to 40, so mas ibig sabihin we have a lot of PCR product or yung ating na-copy na DNA. Okay. So that amount of DNA strand, enough na yun to visualize the DNA or yung products under electrophoresis. Okay. So, anong important component? Of course, we have the primer. We need to design primer. We have forward primer and reverse primer. So yung forward primer will be ito yung 5 going to 3, and yung complementary of forward primer, of course, we have the antisense ng DNA strand natin. So yung antisense, that is 3 going to 5. So 3 going to 5, of course, yung complementary niya is 5 going to 3. And the 3 here has a OH part, which is important part para makatalyze ni DNA polymerase yung addition ng bago nating nucleotide. Okay. So, etong 5 going to 3, forward primer natin ay didikit siya dito sa part na to. Okay? After nating ma-separate tong DNA strand by denaturation. Okay? And of course, yung reverse primer, doon naman siya mag-a-attach doon sa sense strand. So yung sense strand natin, itong 5 going to 3. Okay? So, what else? So, yung PCR or polymerase chain reaction, there are 4 important components. Of course, you have the DNA template molecule. So important yun so sa reaction. And then we need to make primer or two synthetic primers, yung forward and reverse primer. And we need, of course, synthetic deoxynucleotide dun sa ating PCR tube. Of course, itong nucleotides will be used by the DNA polymerase or Taq polymerase dun sa reaction. Okay? And of course, yeah, na-mention ko na, we have the Taq polymerase or the DNA polymerase enzyme. And of course, you have the mix buffer. Okay? So yung mixed water, ito yung aqueous solution natin na important like pH condition, um, um, mixed buffer na mag-aalaw for our Taq polymerase na i-process yung entire reaction. Okay? So ito alam niyo na itong denaturation, annealing, and extension. And as you can see, extension occurs like around 72 degrees Celsius. Yung annealing natin, ito somehow between 50 to 60, depende sa primer na dinesign nyo. So during annealing and during denaturation, we increase the temperature till 95 degrees until such time we destroy all the hydrogen bonds. So meron na tayong separated DNA after denaturation. And then such time, we lower the temperature para kumabit tong ating primer. So yung primer, didikit siya here. Okay? And pag tumikit yung primer natin dito, ibig sabihin may hydrogen bond between the primer tsaka yung maliit na part ng ating target gene or target sequence. Okay? And then after that, yung extension, we try to increase again the temperature to for our Taq polymerase or DNA polymerase enzyme act dito sa part na to, end of ng ating primer, to add more DNA nucleotides. So, first question. So, bakit Taq polymerase yung ginagamit natin? Normally, in our body, during DNA replication, hindi naman umaabot ng 72 degrees Celsius yung temperature inside the cell. So why is it na itong PCR or polymerase chain reaction try to use Taq polymerase rather than our or human DNA polymerase? So yun yung first question sa inyong homework. Okay. So ano yung RT-PCR? So kung meron tayong polymerase chain reaction which amplifies the DNA, the RT-PCR is yung we could try to convert the total RNA or mRNA isolated in the cell or tissue to make cDNA or yung ating copy DNA. Okay, so normally, if we want to know the reaction inside the cell or malaman natin yung kung yung kung kung nag-translate or na-transcribe. Okay, kung na-transcribe or na-translate yung gene, we need to isolate mRNA. So anong ibig sabihin nun? So, for example, meron tayong keratin gene. Okay? So, yung DNA natin all over the body ay pare-pareho. Ibig sabihin, there's a keratin gene for the skin. Meron din keratin gene dun sa ating muscle. may keratin gene dun sa bone. Okay? But to determine if yung keratin gene is na transcribe or na-translate, we need to isolate mRNA from the cell or yung source natin. Okay? So it means mRNA or isolation of mRNA somehow determines if natra-transcribe yung gene. Okay? Kung natra-transcribe yung gene. Okay? So therefore, we need to isolate mRNA or total mRNA, and after isolating total mRNA or RNA, we need to convert that into DNA. Okay? Yung copy DNA. And we call that reverse transcriptase, PCR. Okay. And after we have the cDNA, pwede na tayong mag-PCR. Okay. So, ngayon, nag-evolve na yung technology. We have one-step RT-PCR, RT-qPCR, and another one, we have two-step RT-qPCR. So anong difference? So for one step, after we isolate RNA, we could put the RNA dun na sa tube. And then sa tube, magkakaroon tayo ng reverse transcription. Pag reverse transcription, we convert this RNA to cDNA, or we use this RNA to have cDNA or yung copy DNA. And then followed by qPCR in the same tube. But for two-step RT-qPCR, we need RNA extraction, and then we need to convert this RNA to cDNA by reverse transcription. And then we need another tube to make a PCR reaction. Okay, so that's why we have cDNA to DNA. So, second question. Anong benefits ng one step? Anong advantage ng one step and two step? And ano naman yung mga disadvantage ng one step and two step RT qPCR? Okay? So, okay. So, ang gagawin natin today, we have one step RT-PCR. I will explain the mechanism ng RT-PCR. And the good thing about doing experiment, everything prepared na kasi kit. Okay? Dahil meron ng kit. So, here, meron tayong tube. These are lyophilized, so wala pa tong water. Okay, and inside the tube, we have yung enzyme needed for the reaction. So we have the lyophilized one-step RT-PCR premix. So ito yung nakita niyo yung color blue. This is the premix and dry to. It's a lyophilized. And then, maglalagay tayo ng, of course, ng ating water. We use itong water, RNAs and DNAs, free water. And ano pang kailangan natin? At kailangan natin maglagay ng primers. So since yung samples ko ay virus, so later, later ko na ilalagay yung sample at the end of pag nahalo ko na lahat ng water at ng primer dito sa tube. Okay? So, gagawin ko siya inside the hood pag maglalagay na ako ng virus. Okay? So, yeah. So, ang gagawin ko muna ay mag-label muna ako ng tubes. Okay? Okay. So, ang gagawin natin today, we have So this is me labeling the tubes.
Okay, so tapos na mag-label. Medyo kakaiba yung trip ko talaga sa buhay. So, usually ginagamit ko tong tip box. Lalagyan na ng tip box para doon ko ilagay yung sample ko. Para meron na rin akong ice. So, on ice na rin siya. So, yung reaction volume natin ay 30 microliters at maglalagay tayo ng 2 microliters ng primer, forward and reverse primer, both na yun. At maglalagay din tayo ng RNA natin. Of course, yung RNA or natin ay galing dun sa serum. So, maglalagay ako ng 2 microliters of RNA. So, like 30 minus 4, that's 26. So, 26 microliters of water yung ilalagay natin. So, ang maganda sa molecular biology, sobrang dali na lang ng experiment. Yeah, madaling mag-experiment dahil provided na lahat ng gamit or kit na siya. So parang ipa-follow na lang natin yung kit. Then the idea behind or yung mechanism ng experiment na to, explain ko sa next video. So, kuha lang tayo ng water. And ilalagay natin sa dun sa sample, sa tube. Sorry. So, kung napansin nyo, iba na yung color niya, mas intense na yung color blue niya compared dun sa katabi dahil na-dissolve na nung water. So again, these are lyophilized mixture. So ni-remove lang yung water. That's why we need to add water to rehydrate it. Okay? So maglalagay lang ako ng water and after ko maglagay ng water, maglalagay din ako ng primer. Okay, and explain ko next video yung mechanism, bakit may reaction, anong composition ng reaction, mixture, at ano yung primer, like ano yung forward and reverse primer. Yeah.
Okay, so medyo makulit ako. So again, yung forward primer, that is 5 going to 3, and that will be attached to the antisense strand, itong 3 going to 5, forming a 5 going to 3 strand of your DNA. Okay, and then yung reverse primer naman, siya yung magka-attach dun sa sense strand of the DNA, which is 5 going to 3, so making 5 to 3 direction. So, ito yung gagawa ng 3 going to 5 DNA. Okay? And of course, again, yung nasa loob ng tube, we already have the Taq polymerase, we have the DNA, ay wala pa tayong DNA, hindi ko pa nilalagay yung DNA. We have the Taq polymerase, we also have the mix buffer and nucleotide. So those are the component of our PCR tubes. Dahil lyophilized siya, yung water na inad natin yung magdi-dissolve ng buffer and nucleotides together with Taq polymerase. Okay.